Okay, folks, so this is my slot on the genetic analysis and bioinformatics module. And what I'm going to be talking about is something which we've been researching. It's based around some work we did starting 20 years ago. And it came out of an MSc ABT project. At the time, the BBC were doing this series called, it's about the Vikings, Blood of Vikings. And they were initially working with University College London and a group there who were using genetic methods based around what's called the male Y chromosome to look for ancestry in the current population of the British Isles, to answer the question, when the Vikings came all those years ago, did they leave leave signals behind in terms of the genetics of the present population? And they teamed up. It was BBC with University College London, and they were looking for people to join in and help. And at the time, we thought this might be a good idea. To maybe one of our project students could be involved. So we got in contact with the BBC and Professor David Goldstein at University College London, and they were enthusiastic and took two of our students on. So that's how we got involved, and then that continued after that series. So you must have been about 8 or 9 or so when that series came out. Maybe your parents will remember seeing that. So this came from that, really, and with their own interest in DNA, as you know, from the Crete and all that sort of stuff. This seemed to be quite interesting. So this presentation really is about the use of DNA methods to probe ancestry, either oh gosh, sorry about this. Be careful with the mouse key. Either in terms of populations or in terms of individual ancestry, and this is where the commercial DNA testing comes in, which is now very, very big business, especially in America, who are always after finding out. Americans are always interested in finding out their roots and things, but also across the globe. So yeah, that's what we're going to be talking about: the use of DNA methods for researching population and individual ancestry, and a bit about the role of commercial DNA testers, which has not been absent from controversy over the years. We've seen companies appear and go, but as databases have increased, now the information coming out is becoming increasingly reliable. Okay, so as I said, our interest in this started as an MSc ABT industrial placement, 2001. So this was the early years of the ABT course. And this is Emma, actually. She's standing on a corner of a street in West Kirby, testing one of the volunteers for the Blood of Vikings series. Okay, so what you saw there was Emma and Giles. They were ABT students all those years ago, taking DNA samples from people. And this is back in 2001. And when this sort of testing first came out, people were quite reluctant to come forward because they were a bit curious as to what you're going to do with the data. Give it to the police, or some people even believe you want to clone them and things with all this sort of stuff. And a bit frightened, is it going to hurt? But all you do is just get one of these little brushes. Anyone done a DNA test? It's a bit like a COVID test in a way, except you don't stick it up your nose or down your throat. So you get one of these little brushes, and you actually put it in your mouth. You rub inside your cheek, that side, and then that side. And then when you're done, again, it's a bit like a COVID test. You put your sample, your brush in there, twizzle it about, and then that transfers the material to basically detergent liquid, which preserves the DNA. Then off it goes to the lab. When we were doing this, we were trying to make sure people didn't sort of brush their teeth and things. We say, well, if you do that, you'll trace yourself back to a bacteria or something. So it was quite good fun in the early days because it was all new. But now, of course, it's changed. There's no resistance to this sort of testing. People know what it's about, and they pay good money to. No, you paid, or someone bought it for you.
I think.
Yeah, standard test now costs about £100 to do, but it's interesting, and yeah. There's all sorts of ethics involved with this type of testing. Right, let's come out of this then. So let's stop sharing that. And then we share screen again. Go back to the right. So we're back with this again. Let's go. So DNA. DNA, it's not there so we can probe our past. DNA is there because it codes for what we are. You all know that because you're all been through this. So the main purpose of DNA, of course, the real purpose in nature is it codes for proteins, and proteins. And then on the rest of sort of life takes place. Proteins, enzymes help to make polysaccharides and other structural materials and things. So that's the reason why we have DNA. But because we inherit our DNA from our mums and dads and grandfathers, grandmothers, and going back and back and back, it's passed along from generations. Therefore, it contains a message from our ancestors. So it's a way of unlocking the past. And the last 20, 30, 40 years, methods have been developed to allow us to do just that. And we've been through all this with Michael Creed. That's, Nottingham has obviously a claim of interest to all this sort of stuff. So it provides a tool for individual ancestry, paternity testing, things like that, but also a tool for population ancestry, as we will see. And I don't know which any of these names of companies.
I think I used Ancestry.
Ancestry.com, yeah, or .co.uk seems to be the popular one these days. But these are some of the commercial DNA testers that have been involved. Some no longer exist. For example, Oxford Ancestors has closed, and Britain's DNA also closed under a shroud of controversy concerning false claims, overclaiming what people's ancestry is or are. This was National Geographic. I think this is also closed. This was very good. Decode Genetics has been around for a long time, nearly 20 years. That's based in Iceland. But MyHeritage, Ancestry, and Family Tree DNA are some of the common ones. And Family Tree DNA is also very good. But these DNA testers are as good as their databases. So as databases increase, the method becomes more and more powerful. In terms of population ancestry, this is where we got involved, because after the Blood of Vikings was like a first stab at this in the British Isles, and tended to focus on rural areas, testing modern population, modern people from present day people from various areas in the United Kingdom, but British Isles, but tending to focus on rural areas that have not undergone a large population change or increase over the last few generations. And one area that wasn't really covered in the Blood of Vikings was this area, which Sylvia and myself will recognise, Wirral and West Lancashire, for a simple reason that Liverpool, because of the growth of Liverpool as a port, the population has increased enormously over the last few hundred years. So any attempts to try and look for traces of Vikings coming into this area will be very difficult in that sense. And it was frustrating because we know from all the place names in the area that on both sides of the River Mersey, that this was an area which was settled by Scandinavians all those years ago, including Skelmersdale and Ormskirk, Sylvia, Shalmersdal, Shalmers Valley, and Ormskirk, the Church of Orm. Yeah, but the areas, as you'll see in a minute, full of Scandinavian place names. So it's there. But how can we use these modern genetic methods to try and get around this great population influx that happened more recently when Liverpool grew as a port? And that's when we approached the University of Leicester, because that's the birthplace of forensic genetics. That's where Professor Sir Alex Jeffreys, developed all the forensic DNA methods just down the road from us, 15 miles, 16 miles or so. So we teamed up with them, and and this was the result. We had a paper that was published in 2008, and a book that came out a couple of years later. But I'll say more about that later on. This is Mark Jobling and Turi King from the Leicester team. This is when we were in Norway getting controlled data. Mark is still Professor of Population Genetics at the University of Leicester. Turi was then a PhD student. Now she's become very famous because she was the scientist, after she got her PhD, who did the work on the DNA of Richard III. The final piece of the jigsaw, trying to work out if the guy found in that car park really was Richard III. Then it was the DNA method based on mitochondrial DNA, which clinched that. And Turi was the lead scientist behind that. And now she's famous. She's appeared. She has a TV series where who did she co-host the series with? It's about personal ancestry and trying to find links for people who long lost relatives, or this sort of thing. Stacey, what's the surname? It was Stacey Dooley, might have been. Yeah, she does that series with Stacey. But this was taken in Norway. This is actually in Haugesund, and it's by a very attractive statue. Anyone recognise who that statue is? Some people think it's the mermaid, but that's in Copenhagen. Have you seen the mermaid in Copenhagen? It's actually a statue of Marilyn Monroe, believe it or not. because Haugesund, Marilyn Monroe's real name was Mortensen, Marilyn Mortensen, and a parents, or at least a father, came from Haugesund. And, the Haugesund people decided to put the statue up. We found it anyway, so it's a slide. So what happens to your sample when you've done this, put it into the Eppendorf and then sent it off to the laboratory? So this is Turi and Pila analysing the sample, extracting the sample. So as you can see, it's no laughing matter. And the sample is then transferred to a PCR instrument. I don't like using the word machine, but you've used PCRs, I guess, with Jan. Yeah, as part of this module or as part of the BLT module.
Yeah, we didn't. We set it up. We didn't actually do the PCR, so they just did it for us.
All right. Well, you handed the sample and they put it through.
Yeah.
And so, did she cover the PCR process? The different stages, the denaturation, the annealing using initial primer, using the appropriate polymerase to do the catalysis and the extension. So basically the interesting part of the DNA which we want to investigate with the appropriate polymerases is magnified, so you have sufficient material in order to do the analysis. And this is not real data, by the way, but this is just a slide to illustrate that people have different profiles, as shown by this so-called DNA fingerprint or electrophoretrogram. So the question is from this: are Mr. Hyde and Dr. Jekyll the same person? No. Okay. But this, as a stress, this is not real data. Okay. You've heard of, the Jekyll and Hyde novel. Okay. Right. And they are the same person, really. Okay. So you know all this. This is a slide we generally use for the general public when we are giving lectures on this. So there's a nucleus, and the nucleus contains cell chromosomes, chromosomal DNA, and where the DNA is packaged into chromosomes. And there's 23 pairs in humans. You know all this. And you've got 22 identical pairs, and then pairs that look the same. And then you've got the sex chromosomes. In females, it's 2 X's, and in males, it's 1 X and 1 Y. But what makes this Y chromosome so interesting from an ancestral point of view is that with all the other DNA in the what's called autosomal DNA, the information is shuffled between generation and generation, whereas the DNA, most of the DNA on the Y chromosome is passed along the paternal line with little or no change. So the father passes the Y chromosome DNA without change to his son, and it gets passed along the son to son to son to son, all the way along the male line. The only changes occur where there's a mutation. So a rare mutation will change the sequence, but otherwise it's passed down without change. So that's why the Y chromosome is interesting, because it's a way into the past. Father. So if you can check the Y chromosome type of someone here now, we know what that was like generations into the past. But there's some other DNA, that's mitochondrial DNA. And mitochondrial DNA is passed along the maternal line with no change. So from mother to her daughter, but also her son. So the son will also get maternal mitochondrial DNA, but the son is not able to pass it on himself. So it's passed along the maternal line from mother to daughter to daughter to daughter. So this, if you like, is a parallel probe into the past along the maternal line. It was actually maternal mitochondrial DNA which was used to help clinch the Richard III story via descendants from Richard III's sister. So that's maternally inherited DNA. So this is how it works. You've got the son inherits the Y chromosome DNA from his father's line, and also the mitochondrial DNA from the maternal line, whereas the daughter only inherits the mitochondrial maternal DNA, amongst all the other DNA, of course, from the autosomal contributions from all the ancestors. But we can trace, for a male, you can trace the Y chromosome maternal lines, and for a female, you can trace the maternal line. But the Y chromosome line, that comes with it a huge advantage. I'm sorry, Sylvia, I'm not trying to be male biased here, but and that helps a great deal actually when we're looking at population ancestry. This link, which I'll show you in a minute. Why is that not advancing? There we are. So show you how it works. This is the Hardings, not Tom from yesterday, obviously, but he's not closely related. This picture was actually taken from the station pub, which used to be, you know, where the bridge is on the way to Kegworth. There's some posh houses there now, but that used to be where the railway station used to be, and there was a pub attached to that. This was taken just before it closed. So this is me here. This is my wife, Anne, who's a Leicester girl. And then our four lads, Tom, not the same Tom as yesterday, also called Tom, John, Matt, and Richard. So the lads will have the mitochondrial DNA from Anne, from Leicester, and hopefully we'll have the Y chromosomal DNA from me. So I'd pass my Y chromosome onto the lads. But Anne's mitochondria won't be passed on. So that's the end of the line as far as Anne's mitochondrial DNA is, but I can pass mine on. Now, since that time, John, how does it do that? Has produced a wonderful granddaughter. This is Annabelle. Let's say cheese. Annabelle. This is taken, she's grown up now. She's actually 12, Annabelle. But this is Annabelle. And obviously John is the dad, but the mother is Simone, who's from Germany, from Dresden. So this is the end of the line for me. So Annabelle won't have my Y chromosome because she's a girl. She also won't have Anne's mitochondrial DNA because that will come from Simone. So she's got a German mitochondrial DNA. And similarly with Richard. Richard's my youngest son. And this is Richard, another granddaughter. This is Alice. This was taken last year. And again, this is the end of the line for me because Alice is a girl. And also for Anne because Alice would have got her mitochondrial DNA from Emma, who's French. Okay, so this is taken in Grenoble. But the good news is, from my point of view, is that Alice now has a little brother called Louis, and so he will have the same Y chromosome as Richard and hopefully me as well. So it continues, and that's how it goes. But when you get this, when it stops, because of no more boys and things, or if you produce lots of boys, and so your DNA is magnified, this is called what we call genetic drift when it comes to population ancestry. So it's a factor we need to take into account if lines stop or lines expand because of the numbers of boys that come out. So that's how it works. And so for men, we can look for two types of variations in the Y DNA. The first type is when you get a mutation of a base. Now, this happens only once. Any particular point on the enormous DNA sequence, it's virtually impossible that lightning strikes twice in the same place. So when you get a base change like here, so A is the normal base, it's changed to a T here, that's what we call a single nucleotide polymorphism. So the chances are it will never happen again in that particular place. It's rare anyway. These changes can take place over a course of a million years. So single nucleotide polymorphism. So these are rare events. So this determines what's called a haplogroup, the positions of these single point mutations. You get mutations elsewhere on the chain, okay, but not at the same place. But what's more common is you can get variations in these repeat sequences, which are called short tandem repeats. So these sort of changes can take place over hundreds of years, or even less frequently over decades. So this is a more common variation in DNA by these short tandem repeats. This leads to a man's haplotype, Y chromosomal haplotype. And when you do the test using PCR and then the analysis that follows that, we test for the haplogroup depending on the particular single nucleotide polymorphisms, and also the haplotype. Now the haplotype, these numbers here, refers to the different types of repeats in different locations by these codes here. These can occur, as I said, reasonably frequently. So this is not really useful for deep population ancestry. It's more useful for paternity testing or for near relative analyses and things. But the haplogroup is much more useful for deep ancestry. And this is sort of variation you get with the main haplogroups across the globe. So these variations are caused by these rare single nucleotide polymorphisms, and we've got these are the major haplogroups, and you can see how they're distributed differently across the globe. So Africa got A, I think, for Adam. I think that's just that's what they say. But in Western Europe, it's largely haplogroup R, the pinky, and the sort of duck egg blue, which is haplogroup I. So these are the main large haplogroups in Europe, but different from across the globe. You see how they're grouped and how they change as you move across. But within each haplogroup, you can get other SNPs occurring elsewhere along the DNA. So the first one is like that one there, that defines the main haplogroup. But then later on, you can get other changes along the DNA sequence. And this gives rise to sub-haplogroups within haplogroup R, within haplogroup I or J or whatever. So these are called sub-haplogroups or subclades. And this gives us greater resolution in terms of working out where the Y chromosome may have come from. So let's get rid of this. Away. Right, so this is the percentage of men with haplogroup R. So R subgroup 1b S145. Don't ask me to explain what these numbers mean. So R1b S145, and this is what we call a so-called Celtic haplogroup, and you can see why, because the main density seems to come from Ireland. This was where the mutation may have taken place or likely taken place, and as you go away from there, you can see the frequency gets less and less and less. This is what so-called Germanic or Doggerbank, we call it, haplogroup. Doggerbank for obvious reasons. This is the Doggerbank here. So you can see the largest proportion of people with this group are in this sort of region here, and as you go away, it gets less and less frequent. Then there's the so-called Scandic type, I1, which seems to have arisen from here, from central Scandinavia, where 40% of men have this haplogroup. As you go away, it gets less and less. So you can see where this is leading. Now, in terms of the mitochondrial haplogroup, this unfortunately is not so resolving because the size of the DNA molecule, or the size of the mitochondrial genome, as it were, is much smaller. So that means the variation is a lot less. So haplogroup H, you can see, is quite widespread throughout Western Europe. But again, you can get sub haplogroups. So within H, for Norway, most people have haplogroup H. But within that, you get these sub haplogroup differences as well, and that applies to the other mitochondrial groups. So there is some resolution, but not as great as you get with the Y chromosome. And if you do an ancestry test, so when you did yours, Jack, you must have had it would have told you three things: your Y chromosome type, your mitochondria, depends how much they paid, really. Usually these days it costs about £100, I don't know, £70. You would have got your mitochondrial haplogroup checked, and then you would have got some sort of autosomal check where it gives you so much percentage of this, so much percentage of that. Is that what you got back?
That was the main thing they showed.
Yeah, that's what they base it on, because the main part of the DNA which commercial testers use now is the rest of our DNA, the autosomal DNA, looking for SNPs in the autosomal chromosomes. And that's good for a few generations. I forget what they say now, 5 or 6 generations back, and that's it. And it's as good as the databases on which they do the comparisons with. So I don't know when you had yours tested quite recently, was it?
It was 2018.
Okay. If you got it tested now, you might get a different result because the databases are that much better than they were 4 years ago. But it was interesting.
It was strange. The biggest block that I've had was, I think, 40% Ashkenazi Jewish, which I have absolutely no explanation, and neither does anyone in my family. So I'm not entirely convinced that would be correct.
Yeah, you're right to doubt that, because as I said, it's as good as the databases, and yeah, I would take it with a pinch of salt. More interesting was what your Y chromosome or Y chromosome type was. You can't remember what that was.
I don't think.
Okay, these percentages. So yeah, again, be wary because these. That's one of the reasons why some of these companies have closed, because of these false claims and things. And they should come with a warning. It just gives an idea of your ancestry. Family tree DNA is good. Ancestry, MyHeritage, the National Geographic was very good, but that's run its course now. And Decode Genetics, based in Iceland, is good. Okay, but still, in terms of population ancestry, the male Y chromosome is possibly the strongest, and for good reason. And I'll show you what that reason is now. So, yeah, Y chromosome DNA is still the most powerful marker, in our opinion, because of a very useful property. Do you know what that property is? What else do blokes pass on along the generation besides the Y chromosome? Jack, what's your surname? Wasby. So what's your dad's surname? Your granddad's? And his dad? So I'll repeat the question. What else is passed along the male line with no change? Yeah, Crosby. Not Crosby. Sorry, Jack. Other surnames, yeah, surnames. Of course, there can be non-paternities, adoptions, all this sort of stuff that has to be factored in. But surnames provide a very powerful way in. So this is when we approached Leicester and Mark Jobling and his team. Terry was a PhD student then. Is can we follow up the blood of Vikings and do something which was, had a surname criteria to probe interesting areas, biased, like Wirral in West Lancashire. That was a good choice, I think. Yeah. So that's what we did, and this is show you this. This is some Julian Richards, who's University of York, and this is a map of Scandinavian settlements in England and Wales, and you can see where we are now. Actually, is right in the middle of all this Scandinavian stuff. And actually, if you go from here along the A6006 towards Melton Mowbray, which is along the Reek Valley, you see all these places with BY. BY is a Scandinavian name meaning a village or settlement. Asfordby, Frisby, Barkby. I got married in a place called Thurnby, which is just outside Leicester, Dalby, Freeby. All these places, the whole area is full of Scandinavian names. And Yorkshire, Lincolnshire, East Anglia. These are mainly sort of Danish in origin, because the Danes came over from the east and they settled in great numbers in the so-called Dane law. Whereas the northwest is slightly different, because the northwest was more Norwegian Vikings that came over the Shetlands, Orkneys, down the Irish Sea, and then many settled in the Isle of Man. Of course, the Orkneys, Shetlands, Hebrides. Some went on to Iceland, and then even Greenland and America. But a lot settled in Isle of Man and also Ireland. And then in the early part of the 10th century, Dublin used to be a major Viking sort of trading point until the year 902, when there was a huge battle and they all got driven, or most got driven out. That's when they started coming into the northwest. So that's where that came from. See, so the grey area here corresponds to that. And this is a map, an earlier map, I think by A.H. Smith, of the distribution of place names of Scandinavian origin in England. And you can see it demarks this. This is Danish in the east here, but in the west, you've got the Norwegian settlement. That's not 902, I think, is accepted in the Wirral. So these are parish names of Scandinavian origin, and then in West Lancashire, and also further up in the Lake District. Lake District is also full of Scandinavian names. So that's why we said let's focus on here. We convinced Mark and the gang at Leicester, let's do this, and we agreed that using the surnames to get behind the population movements that have happened since the time of Liverpool growing as a port. And this is the historical evidence behind the Viking settling in the area. These are from ancient Irish chronicles, which report the there we are. That's Hingemund. Ingemund was the leader of the Norsemen who were expelled from Ireland in 902 AD, and it tells how they came to Ethelfled, the queen of the Mercian English. She was the daughter of Alfred the Great, asking Ethelfled for permission to settle in some lands which they weren't so interested in. Now we don't know what sort of deal was set. Some money exchanged. There might have been some romance. We don't know, because Norwegians, Vikings, used to wash at least once a week. Yeah, whereas Anglo-Saxons used to wash, I don't know, maybe never. So there could be some basis behind that, and we don't know. But some deal was set, or Ethelfled may have seen these as friendly Vikings. That let's lock in, and they will stop any unfriendly ones coming in instead. And so the deal was set, and it tells of lands near Chester, which we think was Wirral, because Wirral then was quite marshy and boggy places, and that's probably why the English didn't want it. So okay, you can go there, and then following that, when that got full, it started to go across the Mersey into West Lancashire. Then a lot came over and further up to the Lake District. And that's how the settlements took place, and they're largely peaceful because they were taking areas which the previous people didn't want. And you can see that from the place names so Liverpool's here. These are all names which got Viking roots. Also minor names, Rake, that means a lane. Carr means marsh. And then if you look at all the minor names, you can see the density in Wirral of the settlements. Right in the middle, this little square is Thingwall. There's me with a trammy scarf. Tram is also a Viking name. So there's a story behind that signpost, which I'll tell you about sometime. But there's also a Thingwall in West Lancashire, Thingwall Hall, or South West Lancashire, West Derby. That's where they had their parliament, Thingwall. Thing is a parliament or assembly, and Wall is field. So you've got Thingvellir in Iceland, Tynwald in Isle of Man, Storting, which is where the government in Oslo is. Stor means big, so big thing, the big parliament. That's what Storting means. So we've got one in Wirral, and they've got one you've got one in Lancashire, Thingwall Hall. So this is West Lancashire. These are all the places with Viking roots. Do you know that? You did. Scarisbrick, Leyland. The Norse is Lagerland, actually, which I thought was very appropriate. Lager, not as in beer, but is in low-lying land. Yeah. So there you are. That's how we convinced Mark and Turi. So we all teamed up, and so to get our base of volunteers, we couldn't just test anybody. You had to have a name that was present in the area prior to the growth of Liverpool as a port. So prior to 1600. So to take part, you had to turn away people. I'm Ireland, no, sorry. I'm from Yorkshire, no. I'm Norwegian, no. You have to be either Wirral or West Lancashire. We had a separate set of names for them. For Wirral on our list. And these are based on tax-paying records, Henry VIII's tax-paying records from 1542. Criminal records. You might see someone down there. This guy here. Yeah, he was accused of killing a dog and damaging hedges. Another Thomas Harding, actually, should have told Tom yesterday. Thomas Harding and Robert Poole were found guilty in, I don't know, 1362 or something. Found not guilty. Have to add, found not guilty. So tax records, criminal records, also alehouse records. And in West Lancashire, we had a similar list of people who were paying towards the stipend of the Church of Our Lady at Latham and Ormskirk or something. So those who were paying towards the stipend of the priest. Also, if you had a name that was a place name in the area, like Scarisbrick, like Aldcar, like Helsby, like Upton, like Pensby. If you had a name that was in the, doesn't have to be Viking. These are not Viking names. These are just old names which were present prior to 1600. You could take part in the test. And as far as you knew, your male line came from that area. And that got behind all the population movements. It got behind the great movement during the terrible potato famine in Ireland in the 19th century, when loads of people came over there. And I'm proud to say that Liverpudlians and the people in the area were generally very welcoming to these folks that came who came over and settled. So we were able to get behind those later population movements by doing this. And this is serious stuff. This was at some office in Ormskirk, I forget now where, but we were given this for the day, and did this with the West Lancashire Heritage Association. They were brilliant. So they sort of took over, and they took over the volunteer recruitment. Very strict. People had to show on a form, all these things, and also approve of the test, because there's strong ethics involved with this sort of testing. And you can see how serious these guys were about this. This is, serious stuff. So we got our samples from all these people from West Lancashire and Wirral, did all the analyses, PCR and haplogroup. And we looked at the distributions of the main haplogroup types, and we compared these with data from Scotland, from Ireland, and also from the Orkney, Shetlands, and Norway, and other places, and then looked at the distributions, and then made some analyses based on how similar the populations were to each other in terms of distributions. So what you see here, this lot here, these are modern samples. Okay, so this is from men who didn't, from the area, who could say that their male grandfather was from the area, but didn't necessarily have a surname that was present prior to 1600. And you can see the difference. This is Norway. Okay, Norway's got a lot of this group here, R1b. It's got lots of group I, and lots of this group here called R1A1. Now, these groups appear commonly elsewhere in the region, in Western Europe, but this group here, R1A1, tends to occur in Western Europe only in significant numbers in Norway and areas that have been settled by Norwegian Vikings. So Shetlands and Orkneys, as you'd expect, have also a significant proportion of R1A1, as well as the other groups as well. And Isle of Man and Penrith. And in terms of the modern West Lancashire and Wirral populations, there's a small sign, but not much. So these are people without that criteria of surnames. But when you use a surname criteria, we call these medieval samples. Doesn't mean they're not medieval people, but there's people who've got surnames that were present in the medieval period. You can see how it changes and how the proportion of R1A1 increases quite dramatically. Also interesting is how similar the Wirral and West Lancashire populations are to each other. Different data sets, different surnames, different sets of people, but almost identical in terms of their profiles. This is good because the two sets of data reinforce each other in that sense, and it also shows that the Mersey, River Mersey, hasn't been that sort of barrier over the years, and also ties in with all the place name evidence as well. So that's not too surprising, I suppose. So if you do take this data and then do these population similarity analyses, then you can get an idea of how similar populations are to each other. So let's say the yellow represents, this is not the R1A1 now, the yellow represents British Irish ancestry, and the red represents Norwegian ancestry. Then you can do these what's called admixture analyses, and obviously Shetlands and Orkneys has significant Norwegian, but you can see how significant the Wirral and West Lancashire results are. And if you do the analysis, up to 50% of the DNA admixture, that doesn't mean 50% of the blokes have Scandinavian and the rest don't. It means that the mixture of the DNA is 50% Scandinavian, 50% of Celtic, Anglo-Saxon, or whatever. That's what that means. Obviously, there's error involved too. And the other thing is, if you look at rare surnames, so I'm afraid Smith doesn't come into the but Cromblethwaite, things like this, the names like Totty is another one, Aldcar. These are quite rare surnames in as far as British surnames are concerned. Then the proportion actually increases even further, which reinforces the method. That was published in 2008 in Molecular Biology and Evolution, and you can see the football team of authors involved here. There's Mark, there's me, there's Turi. Ziff was a trainee technician we had over from Denmark during this time. So she hovered between our lab and Mark and Turi's lab. Emma and Giles was Giles. They were the ABT MSc students. Judith Jesh, Professor of Viking Studies in the English department here. Mark Thomas from University College London. Georgina Belden, she was also a PhD student as well. Patrick Waite from the West Lancashire Heritage Association was there. So all working together, all acknowledged. This is what happens with research this type. It involves sort of different disciplines coming in, each lending expertise. Steve Roberts, he was an expert on surnames. So some of the surnames that we had for 13th, 14th century, Joinson and all this sort of stuff, he was able to translate into modern forms. So working together, and with that strict criteria that we had in terms of surname recruitment, refusing people who were outside that criteria, it worked well. One follow-up of that is that when we were doing this 10, 12 years ago, there was this assumption that the R1A1, this yellow haplogroup here, present in Britain, came from Norway. Okay, you also find R1A1 in Eastern Europe, in Ukraine, Poland, Moldova, and even in India, you find R1A1. But since that time, there's been, obviously, the methodology is getting better and better, and there's greater resolution within the R1A1 subhaplogroup. So there's another subhaplogroup, or subhaplogroups of R1A1. So with the latest research, and this was published last year, again the same, Turi, Mark, myself, and some other familiar names there too. We looked at the distributions of these subhaplogroups, different types from different sources: Hungary, Greece, Ukraine, and places. And the main ones we were interested in was GML8, R1A1, GML8, and R1A1, GML9. And with great relief, it was these Norwegian subhaplogroups of R1A1 which occurs in great frequency in northern England. So that seems to vindicate that assumption that we made all those years ago. So yeah, it's nice, and it's nice being a Northwest lad to be involved with that. So yeah, it came out. We did this little book, me, Mark, and Turi, which came out a few years ago. You can buy a copy. It's only a £5. It's, oh, well, I shouldn't say that, it's being recorded, but only a £5 from me. To buy from the shops, you have to pay the full price, so to be upset the publisher and things. But you can get, people can buy it from me. It's limited. I've got a limited number at special reduced price for a £5. And that puts it in a much more sort of readable, sort of layperson sort of language to understand. But yeah, it worked. And yeah, I think working with Leicester, finding the right people to collaborate with, because they had the right pedigree, you know, with Alec Jeffreys, later proven with the Richard III stuff as well. Anyone been to Leicester? Yeah, see you have.
I'm not seeing.
What, see the rugby? You mean?
I didn't know.
Yeah, it's worth going. You know, if you're watching the Tigers or someone play, to go to Greyfriars around that area and see what Leicester have done with it. It's amazing. Yeah, and some people even think that, you know, when Leicester won the Premiership football under Claudio Ranieri a few years ago, some people believed it was something to do with Richard III, because it was a coincidence. It all happened at the same time. Yeah, so that's the commercial and population ancestry DNA story. It is relevant to biomolecular technology. There's some fantastic science that's been behind all this, and it also has strong commercial connotations, as well as you've seen. Yeah, and people pay big money for these tests. Was it your mum and dad?
I think it's my
Your father? Yeah. Did you get tested as well?
I think it's for a bit more.
Oh, right. Okay. I get that retested 4 years on and use a different company.
I was thinking about going with 23andMe, but some of their good test kits are like 120, 130.
Are they? Depends what you ask for, really. But I get the full lot done. Autosomal, mitochondrial. Why? If you're doing it. Yeah, I did it as part of the test. And so did Richard, who got the same result. So we belong to this group here, K-star XR1, or part of this within K-star X R1. So we're only quite small, but you never know. Interesting. It is interesting. It's not a waste of money. I didn't pay for that, by the way. It was good free. What?
My brother did his.
Right. What did he get?
Get back up. Yeah, so we've got all ours in Italy as well.
But he'll have the same mitochondrial DNA as you.
Yeah.
So you get that result for free.
Yeah.
And your dad, of course, have the same result. We hope. It's yeah. Did he pay for that himself, or did? Yeah, that's what happens now. You get this little kit come through. Yeah. Got to send it back and do the test and then mail it off. Yeah, which he did, obviously. Did he ask for it, or was it a surprise? All right.